Hey Team Bio, welcome to your screencast about deoxyribonucleic acid, more commonly known as DNA. We're talking about DNA because it is a very important biomolecule. It is the biomolecule that encodes all of the genetic information of all living things. And we're going to be talking about how the code works in the screencast today and ongoing throughout the whole course, actually. And it's super important because if you understand how to read DNA and what it does, you can manipulate living systems and turn living systems into tiny little biological machines, or cure disease, or create a COVID vaccine. All of these things came about due to our understanding of DNA and how it works. Okay, so the central dogma of biology. States that DNA, which is how genetic information is stored in the cell, is read, and from that DNA, a molecule of RNA is synthesized, and from that RNA, protein is created. And protein leads to the traits and characteristics that living things have. More on this flow later. We're going to be talking about what happens at each of these arrow steps in between DNA to RNA and protein pretty extensively later on in the year. But let's start with this screencast. What is DNA and where do we find it? Well, it's found in the nucleus. Here is an image of a cell. So this is a cell. We know a few things about this cell right away. First of all, we know it's eukaryotic. How do we know that? Well, because it has a nucleus and a nucleolus. These are two structures that we talked about in class the other day. The nucleus is holding the DNA. The nucleolus is the site of ribosome synthesis inside of the nucleus, and it's denser and often darker. It kind of looks like the yolk of an egg. The other reason we know this is a eukaryotic cell is because it has membrane-bound organelles. So here it's pretty hard to see, but this is a mitochondria. The powerhouse of the cell. This is a structure called the endoplasmic reticulum, but we can just abbreviate it and call it the ER. And this is a structure called the Golgi body. More on what all these organelles do later on in the year. So we know it's a eukaryotic cell because it has a nucleus and membrane-bound organelles. And then further than that, we know that it must be either an animal or a protist because it has no cell wall. Okay, so in our eukaryotic cell, we have a nucleus, and inside of that nucleus is a bunch of DNA. The form that DNA takes is at a certain phase in the cell cycle, it tightly compacts and coils into a structure that we call a chromosome. So a chromosome is a coil of coils of DNA and protein. This is the DNA. It looks like string. This is the protein. It's specifically, the protein is called a histone. it's a protein. And the structure of DNA wrapped around this protein, this histone, is called a nucleosome. So a nucleosome is DNA plus protein. Or I guess I should say DNA plus histone specifically. So DNA is it's wrapped around these histones, and then these histones are coiled up into a very particular arrangement, and then those coils are again super coiled on one another into this highly organized structure that we call a chromosome. If we were to take a histone, or a nucleosome I should say, and unwrap the DNA from around the histone, we would see a structure that looks like this. It's a double helix. And a sequence, a specific location on a chromosome, so like this location right here, or this location right here, or this location right here of a section of DNA that we've already unwrapped is a gene. Remember, if we were to take out a chromosome from one of my cells and look at this location, we would find a specific gene. Let's call it gene A. And if we were to take a chromosome out of one of your cells and look at that gene, it would also be gene A. We might have different alleles for at that gene location, but we both have a gene in the same spot on the same chromosome. Okay, so this is a location, this gene. And the sequence of alleles, or the sequence of nucleotides, is the allele. What flavor of gene we have is the allele. And the nucleotides are these little colored things right here. This is an example of one nucleotide. But let's draw the structure of a nucleotide over here as well, because, oh no, it's gonna try to move everything. Never mind, undo. Okay, a nucleotide. What does it look like? Well, it's composed of, oh, that's gonna be a terrible shape. Okay, I want to automatically, yeah, perfect. It's composed of a 5-ringed sugar called a deoxyribose sugar. Oops. That's where the D comes from in DNA, deoxyribose. So this 5-membered sugar, this 5-carbon sugar, is kind of the center of the nucleotide. And then off to one side, we have a group called a phosphate group. And off to the other side, we have what's called a nitrogen base. The nitrogen base comes in 4 different flavors: adenine, thymine, cytosine, and guanine. And I'm going to color them here, specific colors. Color, whoops, that's way too big. Color your A, your adenine, yellow. Your thymine, purple. Your cytosine, red. And your guanine, blue. Okay, so, and this is for a specific reason, because these nitrogen bases, these different flavors of DNA that can be on this side of a nucleotide. We see them all labeled, or not labeled, they're unlabeled right now, but we see them all depicted in this image here. So on this side, I want you to write in red, cytosine, in blue, guanine, in yellow, adenine, and in purple, thymine. Okay, so notice this is a single nucleotide. It's kind of upside down of the one we drew. And it is, so we see an example of guanine bound to cytosine on the other side. And then here we see an example of adenine bound to thymine on the other side. And if you look all the way down this strand of DNA, you'll see that A is always bound to T and C is always bound to G. Regardless of what side the different base pairs are on, we always see C and G and A and T. So this is called specific base pairing of DNA. This is super important because it means that if I only give you one side of a molecule of DNA, you can predict with 100% accuracy what the other side of DNA is going to be. Franklin, no barking. Hey, no, that's enough. Sorry, I'm doing the screencast at home. So this is really important in DNA replication. This molecule is going to split right down the middle, and we're going to synthesize two new sides when the cell divides. Okay, another thing that I want you to notice is that the nitrogen bases are in the center. A is always bound to T, C is always bound to G, and the two sides of the molecule are running anti-parallel to one another. And what I mean by that is that if you look at the sugar group of this nucleotide, you'll see that the phosphate is on top here. But if you look at the sugar group of this nucleotide, the phosphate is up here. So this is what we call anti-parallel. The two strands of DNA are running anti-parallel to one another, and this is called the sugar phosphate backbone. And there's a sugar phosphate backbone on both sides of the molecule. Sorry, the train is going by. You might be able to hear it in the background. Okay, so how does this strand of DNA actually encode genetic information? Well, it's a sequence. What do I want to say? I guess I'll erase these. So this is, if we read the strand of DNA, we see that we have an A followed by a C followed by a G. Oh, you can definitely hear that. That's the train. Followed by an A followed by a T. And the order of A's, C's, T's, and G's is important. It has a meaning. Just like the letters in these two words, A, sequence, have meaning. And if I took the letters and I scrambled them up and I rearranged them, they would mean something else, or they might not mean anything at all. It might just be goobity gluck. The same is true of nucleotides of DNA. The order of the nucleotides is a code. And that's why we call DNA our genetic code. Nucleotide sequence. Okay, so we're going to talk more later on in the year about how the sequence of A's and C's and T's and G's can be decoded and the message that they make and then the little action things that they create that actually do the things of making the traits and characteristics later on. Okay, but I think that's it for today. See you all in class next week.